Hello, and welcome to the webinar on the compatibility of selected organic pesticides and fungicides with biological control. I'm Alice Formiga of eOrganic, and this webinar will last about 45 minutes, and then we'll have 30 minutes for questions. So today, I'd like to welcome back JC Chang, who gave an introductory webinar last year on biological control, which is in our archive of webinar recordings. JC is a professor and extension specialist in the Department of Plant and Environmental Sciences at Clemson University. His program focuses on studying the ecology of pests and developing IPM programs in specialty crops. One of the things that he's been working on is evaluating new and reduced risk products against whiteflies, thrips, and two-spotted spider mites, as well as evaluating the compatibility of new and existing chemicals with biological control agents. So welcome back, JC, and I'm going to turn off my video and hand over the webinar to you.
Thank you, Alice. Thank you, everybody, for attending today's seminar. And it's great to be coming back to eOrganic and talk to everybody again. Well, about something that I have always been working with, basically, is looking at how insecticide will actually impact the survival and also reproduction of biological control agents. So let's get on with that. And all right, so before we get started, I guess I'll talk about, let's do a reminder, a refresher anyway. So what exactly is biological control for those of you that are not familiar with this term? But I figure most of you are. So biological control is basically using the natural enemies of pests to control the pest population. So these natural enemies could be predators, parasites, and also pathogens as well. For example, in this picture is showing is a predatory mite that's feeding on a two-spotted spider mite. So that's biological control. And my mother, who doesn't really have much of a knowledge about entomology, she doesn't really know what I do exactly. So the best way that I can explain to her what biological control is basically good bugs eating bad bugs. That's the easiest way to really think about what biological control is. So for those of you that Alice mentioned earlier, is interested in having more of a background or basic understanding of what biological control is, please visit the webinar that we provided in 2021 about biological control options in vegetable production. In that particular webinar, my postdoc at that time, Goen Gill, and I will be talking about, what are the biocontrol options, what are predators, what are parasites, what are pathogens, and how we can use them in vegetable production. So if you're interested, feel free to go with that. So a lot of the system that I work with now, other than turf and ornamentals, is vegetables. In vegetables, biological control plays a very, very important role. Now, in most of the time, outdoor vegetable production, I do not recommend just buying some biocontrol agents, for example, lady beetles, and release them, because they will just fly away for the most time. So there's not a whole lot of sense into doing augmentative biocontrol, meaning that there's not a whole lot of sense to buy and release biocontrol agents. Buy and release biocontrol agents actually works a lot better if you are using them for controlling pests in the greenhouse, where it is enclosed and the biocontrol agents are not going anywhere. So for outdoor production, oftentimes we're talking about more of a conservation biological control, meaning that we're trying to conserve or protect the natural enemies that are already in the system. So there are a lot of ways of doing that. In fact, on eOrganic website, you're going to find a lot of information about how to use conservation biological control to for pest management. One of the way is actually planting companion plants in amongst your vegetables or your crop. And in this case, it's basically planting sweet alyssum in among the plants and using the nectars and pollen produced by the sweet alyssum to attract a lot of biocontrol agents. For example, circuit flies or aureus or what am I thinking about? Parasitoid. So you can actually attract them. They're attracted by the pollen. They're attracted by the nectars. Once the biocontrol agents have their fill with the nectar and pollen, it's more likely for them to actually spread into the crop around the companion plants or the banker plants to actually control the pest population there. Another way you can do conservation biological control is to provide them with shelter. And beetle bank is a very common example of using some kind of plant to provide shelter for the beetles, for the ground beetles in this case, so that they can stay there during the day, and at night they can spread out to control your pest population. During the day, they come back. So you basically provide them with a shelter to escape from their own predator. So that's a great way to also use conservation biocontrol agent in your crop production system as well. Now, what I'm talking about today is going to be another way that you can do to conserve your natural enemies. And that's it. You to think about how you actually use pesticide in your particular system. You can conserve natural enemies by not using pesticide at all, obviously. But in that case, if you are not using pesticide, you're probably not going to have a whole lot of impact on the natural enemies. Because think about this, pesticide, for example, insecticide, they are designed to kill insects. If you have a ground beetle in the system, that insecticide is designed to kind of kill that ground beetle. So by not using those pesticides, you are basically eliminating that risk to your natural enemies. Another way you can do that is if you have to use pesticide, whether it's insecticide or fungicide or miticide, doesn't matter, is to use a pesticide that is compatible with your natural enemies. So compatible pesticide, you probably heard that term before. And depending on the people you talk to, you really have several different definitions, what compatible pesticide actually is. So in the most general sense, in my mind, there are two definitions for what a compatible pesticide is. The first one is the pesticide, when you mix them together, some kind of chemical reaction happen, and then they either coagulate and then they separate or they become solid or something happen that basically render both or either one of those pesticides ineffective. So when they are not effective, that means they are not compatible. But what we're talking about here is basically physical compatibility. And if you are tank mixing any kind of pesticide at all, I would definitely recommend that you do a jar test just to make sure that the two pesticides that you're going to mix together, or the fungicide and the pesticide you want to mix together, are in fact compatible, that they are not going to interact with each other and reduce the efficacy in any way. But that definition is not what we're going to talk about today. We're not going to talk about physical compatibility. Instead, what we're going to talk about today is compatibility with biological control agents. That's basically pesticide. A compatible pesticide is basically one that doesn't reduce the survival, the foraging efficacy, and also the reproduction of your biological control agents. Okay, so those are the pesticides that have very minimal or no impact on the natural enemies. So when we talk about compatible pesticide, we also talk about, their lethal toxicity. So lethal effect is basically the effect the pesticide have on the survival of the biocontrol agent. The sublethal effect is basically the effect that doesn't directly cause mortality, but they will reduce the foraging efficacy or the reproduction of the biological control agents. So those are just some of the terms that I want to put in your head while we are thinking about this discussion about compatible pesticide. So again, today, in today's webinar, we're just going to talk about compatibility of pesticide with biological control agents. So pesticide, doesn't matter whether they're compatible or not, can be exposed to insects or pests or the biocontrol agent in that matter, two ways. It could be a direct exposure, meaning that the insect or the mites are in contact with the insecticide solution or the dust or the fresh residue of the insecticide directly. That could happen when you spray a pesticide directly on a pest or an insect, or the insect could be getting in contact with a very, very fresh residue of the pesticide immediately after the residue has dried, or that the pesticide could be eating the solution or the dust or the fresh residue, either by themselves or by ingesting the plant tissues that happen. So in this particular situation where the insects are basically in direct contact with those fresh residue or the fresh insecticide very quickly. Residual exposure, on the other hand, we typically consider residual exposure as happening after a few days or after a few hours of pesticide applications. So usually what happens is you apply the pesticide, if it is a spray, they dry, they form a residue, a layer of residue on the leaf surface or the plant surface, and then an insect walk by and they will get in contact with the residue. And once they do that, well, they basically get in contact with the residue or the residual exposure of those pesticides. Or they can ingest plant tissue that actually have those residue on the surface, and then they got poisoned that way. So those are basically two ways as far as how insects and mites can get in contact with pesticide residue. So why is it important for us to think about compatibility with pesticide and biological control agents? Well, it has a lot to do with it because in any integrated pest management program, biological control is a very important component, but at sometimes we might actually need to use pesticide to reduce the pest population as well, either as a rescue treatment or reducing the secondary pest outbreak in some way. So we kind of need to understand how the pesticide that we apply can impact the biocontrol agents that we already have. So if you already have biological control going on, or your natural enemies are already in the system, the information that we get from compatibility of pesticide can help you decide which pesticide you want to use so that you can protect the natural enemies that are already in the system. Or if you are thinking about, if you are growing crops in a greenhouse and you're thinking about releasing biocontrol agents, then you need to think about, well, okay, are the pesticide compatible or not? In that sense, it's basically, it will help you to choose the pesticide that is not going to have a whole lot of impact on the foraging behavior or the survival of the biocontrol agents that you are going to reduce. So it's both now and also in the future. In the future, for the future releases, you kind of need to know how long do you have to wait before you release the biocontrol agents. Because if the fresh residue of the pesticide has very high impact on the survival of the biocontrol agents, you spray it and then you immediately release the biocontrol agents, you're basically killing all the biocontrol agents. That's not going to help whatsoever. But usually for most of the pesticides, there is an effective period. After those periods, the pesticide is probably going to have very minimal impact on the biocontrol agents. So you can actually release the biocontrol agents after a certain period of time after the application. So all this information will really help you decide whether you should release them now, whether you could release them now, whether you could, and how long you should be waiting before you can release them, and what you can use. So that's what compatibility of pesticide would be important to think about when you're doing biological control. So in most of the cases, after we know the compatibility of pesticide, then you know certain pesticide, you should never use them. And those pesticides typically falls in the group of broad spectrum insecticide or fungicide. Usually in that case, things like pyrethroid, organophosphate, and things like that, you should never use them, obviously. And for you guys working with organic agriculture, that they're not going to be used anyway. So not only that, some of the organic or some of the organic pesticide, including ones that's OMRI-listed, could have very significant negative effect on the survival or foraging behavior of the biocontrol agents as well. So we kind of need to know what those are. Some of the pesticide, you can kind of use them. Some of the harmful pesticide can be used if you actually waited long enough that the residue is no longer harmful, you can wait for that. Or you can use a harmful pesticide if you are treating only hotspot, leaving a lot of area that's not treated where the natural enemies can maintain the population or they can hide. So that's how you could actually use those. All right, so there's actually a lot of information that can be found about compatibility information. There's a lot of chemicals out there. There's a lot of fungicide. There's a lot of insecticide, and a lot of miticide. There's a lot of product out there. So this brain of mine, as big as it is, cannot fill all that information. There's no way. So the good thing is there are actually resources out there to help us figure out the compatibility between pesticide and also biological control agents. One of these is, this is one of those. We usually call this the side effect manual or side effect database. Side effect is basically the lethal or sublethal effect that you may have on the biocontrol agents. So this is from the company called BioBest. Some of you might have used that before. So on one side, you can choose whichever pesticide you will be using. And the right-hand side, you can choose whatever biological control agents that you will be using. So the biocontrol agents oftentimes are the one that is produced for release. It doesn't include everything. That's impossible. But oftentimes what I would suggest is that, think about the grouping of those biocontrol agents. You cannot get perfect information about the species that you are most interested in, but you can get an educated guess by looking for information on a related species. For example, if you have a predatory mite that you have no idea what it is, what species it is, but you might be able to find out whether some of the pesticide that you plan on using is compatible with this particular natural enemy species that you have in your system by looking at information about the other predatory mite species. Okay, so you could choose the information, you can choose the pesticide, and then you can choose the beneficial organism, and then the database would basically tell you what kind of toxicity they may be. And in this case, for the BioBest database, they rank everything from 1 to 4. And these numbers basically represent something very important. And oftentimes, these are direct or lethal effect of those insecticides. For example, number 1 is considered non-toxic, meaning that the mortality of that product for the particular biocontrol agent is less than 25%. Number 2 is slightly toxic. That's 25 to 50% mortality. Number 3 is moderately toxic, 50 to 75% mortality. And number 4 is toxic, which is more than 75% mortality. Typically, I think a pesticide is compatible if it's non-toxic, meaning that it's number 1. And it is still compatible if it's number 2, but you might want to think about how you release them, when you release them, and whether you need to create some kind of refuge for those natural enemies. If it's number 3 and number 4, I'll be very careful about using those. You only use them if you have to. And you have to think about their residual longevity. And if you're going to re-release the biocontrol agents again, you might have to think about how long that residue is going to last so that you can time when you want to release the biocontrol agent. Okay, anyway, this is one of the database that's freely available to everybody, is BioBest. Another big company, Cobert, also have their own system. And in this case, I kind of did a little play with it. So I look at the compatibility of azadirachtin, pyrethrin, and spinosad against Ambrosia swirsky. Ambrosia swirsky is pretty common used predatory mite for controlling whiteflies and thrips. All right, so in this case, the system basically spit out the information as, okay, azadirachtin for the adult is probably non-toxic or compatible, but for the entire population, which include adults and nymphs and eggs and everybody, you might see a little bit of reduction in the population. So, I might want to think about, okay, if I'm using azadirachtin and I already have Swirsky, I probably want to watch the population just to make sure that, everything is fine and dandy. Pyrethrin, on the other hand, there's no information. That's another point that I really want to bring out, which is that we have so many pesticides that's registered for organic production, but not every product has the information available for the biocontrol agents that we're interested in. So do not be too surprised when you actually go to this kind of database and click in all the information, and then you'll find no useful information. Sometimes that happens. So unfortunately, in that case, you should probably talk to your biocontrol agent supplier or your consultant or your local extension agents or specialists just to get their takes on, whether it is okay to use that product or not. So more conversation should follow, and just so that you have the confidence in knowing that the decision you are making is a good one. All right. Anyway, for Ambrosia Swirsky, Spinoza, again, that's no bueno because it's number 4 for both eggs and adults. So if you have Ambrosia Swirsky that you use for biological control, you probably do not want to spray Spinoza because it will wipe out your population. That's not going to be great. But again, it doesn't mean that Spinoza cannot be used. You just need to think more carefully about using it, because you can find information about the residual longevity of the spinosad, knowing that if you use a spinosad now, you're going to wipe out your natural enemy population. But you can wait until the residue toxicity has reduced, then you can release them again, or find a way to entice them back into the system again. So like I said earlier, even product that's number 4, there are ways to actually use those without having a huge impact on the biological control program. So anyway, let's talk about something that we have done in our lab as part of the ORII grant. ORII is the Organic Research and Extension Initiative project. So in this particular project, we look at how to really use biological control, or how to encourage the use of biological control in the vegetable production system in the South. So one of the things that vegetable growers always wonder is, all right, I know that when I grow vegetable, and if I grow companion plants, I'm going to attract a lot of things coming into my system. But sometimes I really need to use a OMRI-listed product to actually reduce some other pest population. So what would happen if I use those products? We always get questions like that. So the objective of this particular study really is to provide some information as to the what if. So in this particular study, what we did was basically selected 9 different commonly used OMRI-listed insecticide and fungicide, and then look at their toxicity, or look at their direct contact impact on 7-8 different kind of biological control agents. Okay. So the list is long. To make it easy, I'm just going to go through this particular product and also biocontrol agents, so that you have a little bit understanding of what they are and why we're choosing them. So the first one is azadirachtin, and in this case, we use ecoGarden as one of our examples. So ecoGarden has 1.2% azadirachtin. The IRAC code, which is the Insecticide Resistant Action Committee code, is unknown. We don't exactly know how it works. Now, when you do a research on azadirachtin, you'll probably hear something about, the mode of action is repellent, and it's shown up on the ecoGarden label. It can be a repellent. It's also an anti-feeding, and also an insect growth regulator. So what is it exactly? Well, azadirachtin turns out to be one of those that have all of them. We don't really know exactly how azadirachtin work, even though we've been using azadirachtin for more than 1,000 years. But it doesn't really matter. Even now, we don't really know what it is. Maybe that it has multiple mode of action possible. But anyway, azadirachtin, as far as the products are concerned, a broad spectrum. You can use it for a lot of different pests. And we know a little bit about how azadirachtin is whether it's compatible or not with biological control. So in this particular case, I'm just going to show you, and also the following slide, I'm going to show you the non-compatibility of particular product, a particular active ingredient. I'm not talking specifically about product, I'm talking about the active ingredient. And these are basically for groups of either predatory insects or predatory mites or parasitoid in general. I'm not thinking about particular species. And also these are compatibility by spraying, doesn't include drench. Okay, so a little bit caveat there. Anyway, for azadirachtin, the compatibility that we know for the predatory insects is pretty harmless. Most of the study basically showing that azadirachtin is quite compatible with predatory insects. For predatory mite, is either harmless or moderately harmful in some cases. For parasitoid, parasitic wasps, they are harmless as well. Dipel, which is another product that we use, and most of you are probably quite familiar, is Bacillus thuringiensis Kurzbach. It's 54% of those. The IRAC code is 11. And as you know, dipel, we only use it to control caterpillars and caterpillars only. So if you try to use it to control Colorado potato beetles, you're probably going to pick another product. Anyway, for for BT, we know the compatibility relatively well. For predatory insects, it's typically harmless. For predatory mite, it's harmless, and for parasitoid, it's harmless. So BT, because they are so specific, they only impact caterpillars, and they only impact the caterpillars when the caterpillars eat the bacterial spores. It is not going to impact any other insects or mites or parasitoid, unless your biological control agent is actually a caterpillar, which there's none. Okay, so typically dipel is a very compatible product with biological control agent. The next insecticide that we look at is Grandivo. Grandivo is also a bacteria product. It's a Chromobacterium subsugate. And Grandivo is used for control of a lot of different species, particularly aphids and caterpillars, mites, thrips, and whiteflies. Those are used quite a bit. Again, they are bacterial. And we do not know much about the compatibility of Grandivo. So most of the research papers that I go through seems to suggest that Grandivo is compatible with biological control agents. But in general, we need a lot more information about this particular insecticide. Organic, you guys are probably quite familiar with pyrethrin. Organic is just one of those examples, and this is 3A. So pyrethrin is a broad spectrum insecticide. It can kill a lot of insects very quickly as well. And therefore, it is not really surprising to see that the non-compatibility of pyrethrin in general are harmful to a lot of predatory mites and parasitoids. There's not a whole lot of information available for predatory insects, but my feeling is they're probably going to be quite harmful for predatory insects as well. Right? Spinosad or Entrust. You guys probably are familiar with this as well. IRAC code is 5. Typically, Entrust is or Spinosad is quite effective against caterpillars and thrips, and also leaf miners as well. If you have fire ants in the field, you could also use that to manage fire ant species. In general, Spinosad is actually harmful to predatory insects, predatory mites, and parasitoid as well. So for most of the information that we have seems to be suggesting that, Spinosad is probably something that we need to watch if we're going to use it for in conjunction with biological control. So let's look at some of the fungicides. In this case, one of the products that we look at is Serenade. Serenade, the active ingredient is also a bacteria, Bacillus subtilis. The FRAC code is 44, and it has it's a broad spectrum control of a lot of fungal and also bacterial diseases as well. And again, we don't know anything about, well, we know very, very little. We cannot get any kind of conclusion at all on whether Bacillus subtilis is actually compatible with predatory insects, predatory mite, or parasitoid or not. So again, another goal for this project is to fill this knowledge gap, just to find out whether they can be used or not. Coside is 60.1% copper hydroxide. It also has a broad spectrum suppression of fungal, bacterial, and also algal diseases. We do not know how well copper hydroxide would actually be compatible with predatory insects, predatory mite, and also with parasitoid. So again, we need to find out more about this. Nortox, some of you probably use this as well. This is another copper fungicide, cuprous oxide or copper oxide. In this case, Nortox is actually used for broad spectrum control of fungal and bacterial diseases. And we don't really know much about predatory insects and parasitoid, but there are some information available to suggest that copper oxide is actually quite harmful to predatory mites. But study seems to point to a lot of different directions. Several different studies reporting that it is harmless. Some of them reported that as harmful. So in some way, we want to kind of see for ourselves whether Nortox actually have a high toxicity for some of the biocontrol agents that we have selected or not. And oxidate is basically a combination of hydrogen peroxide and peroxidate acid. It has a broad spectrum against fungal and bacterial diseases. As far as I know, there's not a whole lot of information as far as whether hydrogen peroxide or peroxidate acid are actually compatible with any of the biocontrol agents or insects and mites that we are looking at. So we don't really know how this particular fungicide will actually do. So those are the insecticide and fungicide that we're talking about. Now, let me introduce to you some of the insects and biological control agents that we're looking at. Now, we're only looking at predatory insects and mites because we're kind of focusing on this particular group at this time. In the future, perhaps we'll look at parasitoid, and maybe we'll even look at some biopesticide as well. But right now, we're just going to look at the predatory insects and mites. So there are 4 predatory mites that we look at in this particular study. We have Ambrosius swartzi, we have Ambrosius andersoni, Neostilus californicus, and Phytosteus similis. So each one of those are used to control either spider mites or either fire mites or toxin-emitting mite, or whiteflies and thrips and other pest species as well. So those are the 4 species that we deal with. And we also included in our study Chrysopolla or lacewings. They are typically used for aphids, but a lot of soft-bodied insects as well. So we just want to look at whether they're available or not, whether they're compatible or not. And then we look at a small, tiny little beetle called Delphastus catalini. And this is one of those lady beetle species that we find in the field quite frequently, feeding on whiteflies, both whiteflies eggs and whiteflies nymphs as well. Sometimes if they can catch the adult, they'll feed the adult as well. By the way, this is a very tiny beetle, but they can be quite effective in the field in controlling whitefly population. So we kind of want to look at this particular species as well. And Aureus insidiosus, well, for those of you that have been outside and play with flowers quite a bit, you've probably seen Aureus quite a bit. Aureus or minute pirate bugs or insidious flower bugs, they have been used for biocontrol for many years. And if you grow alyssum and some of the yellow flowering crops, you're probably going to attract a lot of them into the field. So because they are so ubiquitous in our system, we also want to look at their compatibility. Statuarus punctillum, this is another small beetle, but these little beetles actually only feed on spider mite. So it's very much a spider mite specialist. They love spider mite eggs, but they can also feed on the adult and the nymph as well. So those are the 8 species of biocontrol agents that we look at. So I'm just going to go through very quickly as far as how we did the experiment. So all these biological control agents, the lacewings, the aureus, and the predatory mites, and the two lady beetle, the two beetle species, we buy them directly from biological control agent supplier. In our project, we originally wanted to look at different systems and see what are the most common natural enemies and do our testing on those native natural enemies. But unfortunately, in our project, even though we do quite a bit of sampling, we don't see a lot of natural enemies in our system for whatever reason. So as a proxy, we decided to just go with these commercially produced biological control agents. Just again, like I said, give us a little bit of information or proxy as far as how the biocontrol agents are actually going to, whether they're going to be compatible with the pesticide at all. So anyway, we want to make sure that we buy them, they ship it to us, and then we want to make sure that they are looking good. They are not dying crazily. So what we did was to make sure that we check their viability and make sure that they're okay before we start our experiment. And we use them for our experiment within a day of receiving them. So these biocontrol agents are in the top shape as possible. What we typically do is a very simple, a very standard procedure in testing for toxicity for these biocontrol agents. So what we do is we mix the biocontrol of the fungicide or the insecticide, and then we dip. We cut the leaf disc from tomato leaves, basically around this, and then we dip this leaf disc in the fungicide or insecticide solution. We take them out, and then we allow them to dry, and then we put this leaf disc into into a petri dish or a shot 1 ounce shot cups, and depending on what the biocontrol agent wants to eat, we provide them with food so that the biocontrol agent, we know for sure that the biocontrol agent die from contact with the insecticide, not because they starve to death, because we provide them with food. So for some of the species that prefer to feed on spider mite, for example, Androctonum californicus, Assimilus, and Statoris, what we do is we actually put spider mite onto these leaf discs, let them lay eggs, then we put in this biocontrol agent. For species that will feed on other insects, for example, lacewing, Delphastus, and also aureus, we actually buy some flower moth eggs. These come as just basically eggs. We sprinkle some on top of those leaf discs. Then we introduce these biocontrol agents into the shot cup. And for some species, for example, Ambrosius swirsky, they actually will feed on pollen. So for this particular species, we actually put cattail pollen on top of the leaf disc. So in all the cases, what we do is that we provide them with this alternative food. So we know that whatever mortality that we reported is not because of starving, but because they actually die from that contact with the residue. Okay, so keep that in mind. All right, so again, this is the insecticide and fungicide that we tested. So we assume an application volume of 100 gallons per acre. That's somewhat standard for greenhouse application, I think. By the way, the application rate is based on the high rate for vegetables. So as you can see here, most of them are per acre basis, anywhere from 6 fluid ounces all the way to 64 pounds, 25 pounds per acre. So rate vary, but understand that all these rates are really based on the high label rate for vegetable use. And so we after we put all the eggs in there or the pollen and whatnot, we release the biocontrol agents. We put 10 of them in there, and then we just keep them in there for 3 days. 3 days later, we see how many of them die, how many of them are still alive, and also we check to see whether they lay eggs or not, so that we can determine the impact on reproduction. Not only do we do that, after we count the eggs, we remove all the biological control, but keep the eggs inside the shot glass, and we come back 5 days later. So 8 days since we release them, and see how many of those eggs actually hatch, so that we can see whether the impact whether the agent pesticide actually have some kind of impact on it. So for each treatment or each product, we actually replicate it 10 times and analyze the data. So those are the glorious details about experimental design. Don't get nervous about all these graphs and whatnot. This is just a way for me to show you what kind of data that we typically get. In some cases, for example, Californicus, as you can see, the number of dead Californicus in petri dishes treated with, in the shot glass or leaf disc, treated with water only, has fewer number of deaths, which is great. But for a lot of other products, we are seeing some elevated mortality in those cases. This is the numbers of that in Persimilis. Again, water, the number of dead in water treated petri dish is lower. That's good news, meaning that we're not really seeing a whole lot of natural mortality. In some cases, for example, insidiosis, we're not seeing a lot of mortality in other products as well, except for pyrethrin and eco-garden insects. And this is lacewings. Again, mortality in water is pretty low, but for some product, for example, pyrethrin is sky high. So this is how we can actually see whether a product actually have impact on the mortality of the insect or not, by comparing the mortality between the treated and the water treated dishes. These are number of eggs. Like I say, we look at number of eggs, but unfortunately for a lot of species that we work with, they don't produce eggs that well, unfortunately. But in some cases, for example, aureus, they do produce quite a few eggs. So we can get some data to actually do some calculation there. Of course, again, we look at hatching of eggs. So again, I'm not going to go through all those slides because I'm not going to have that much time, but this is basically the summary that we have. All right. So as far as when it comes to killing the biocontrol agents, this is a summary of for each individual species. Whatever active ingredient that I listed on this slide are basically the product that probably should be used more carefully, because in this experiment, we are seeing a significantly elevated mortality compared to water. So I'll come back to this later on with a much more condensed summary as well. But as you can see, earlier when I do the introduction, I was saying that azadirachtin in most cases seems to be showing that they are pretty harmless to a lot of biocontrol agents. But in our experiment, it turns out that azadirachtin actually quite toxic. So is pyrethrin and spinosad as well, and some of the fungicide too. As far as reproduction, meaning that eggs, whether they're with the number of eggs produced or the number of eggs actually hatched. For most of the species, we don't have enough information to actually say one way or the other. So we need a lot more study on that. But for some other species, for example, aureus and insidiosis, we are seeing that eggs that are leaf discs that actually have azadirachtin and pyrethrin, we actually have fewer eggs being produced there, and also the number of eggs that actually hatch are lower as well. So this is basically a very much an overview, 10,000 feet overview of the summary of our result. So one thing that I really need to mention to you is that what we're seeing is that we see a lot of variability among different predator species. So whenever we think about compatibility of pesticide to any kind of insecticide or fungicide, we need to think about this as per species basis. So it's not enough time for me to really go into that today to look at individual species specifically. But in the future, do think about, thinking about each individual situation as a separate situation. So in general, azadirachtin, pyrethrin, and spinosad usually result in pretty high mortality, low egg production, and full egg hatch. So that's issue. Couple fungicide, two of them, Nortox and also Coside, they seem to have pretty negative impact on the survival of the predatory mite. So if we have a lot of predatory mite in our system and we want to control the disease, a couple fungicide may not be the best option in this case because it will reduce the survival of predatory mites. But again, we need to understand the residual toxicity of this fungicide, which we did not touch on in this particular study. Because if we know how long the toxicity is going to last, then we know when we can release those predatory mites again. In general, BT, Chromobacterium or Grandivo, B. subtilis, and also hydrogen peroxide and peracetic acid generally have pretty low impact. So I think this product probably will do well when you use them together with biological control agent. Okay, so this is basically the end of what I have today, and I want to say thanks to Don Shikora, who is my graduate student and worked a lot on getting the data in this particular study. Sean Chandler and also Courtney Gregg, they are my technicians. They also help out quite a bit with the study as well. Lastly, I also want to acknowledge the funding from OREI, or Organic Agriculture Research and Extension Initiative, for their funding to our project in the past few years. All right, so now thank you for attending the webinar, and I'll be happy to answer any questions you might have. So if you have any questions at all, feel free to contact me at that email address, zhuanghc@clemson.edu. Thank you very much.
Okay, thank you so much, JC. That was really interesting. We're getting some questions already, but we certainly have time. So if you have any questions, please type them into the Q&A box, and we'll be reading them out loud in just a moment. I just wanted to remind anybody who wasn't there right at the beginning of the webinar that the recording will be placed on the eOrganic YouTube channel as well as on the eOrganic website at eorganic.org within the next week. And we'll also be posting on eOrganic a handout of the slides in case you want to refer back to them or share this presentation with others. So moving on here to the questions. Let me just pull up my Q&A box here. Okay, and also there are a couple of questions in the chat, so I'll look at both. We had a couple of questions about whether you know anything or studied actinovate fungicide.
No, we did not. That's one of the products that we think about testing, but we never really get around to doing that. So that would be something that we want to look at in the future, for sure.
Okay, great. Do you have any contact on Clonostasis rosea control of fungal diseases?
Well, sorry, Wei Yang, you are out of luck. I'm an entomologist. I do not know a whole lot about fungal diseases. So perhaps we'll have to ask one of our pathologist colleagues on that and get back with you.
Okay, yeah, and JC's email address is on there in case he doesn't remember, so feel free to
Please.
contact him afterwards. Sometimes it's easy to say on these webinars, oh yeah, sure, we'll get back to you, but then, it's kind of good to follow up afterwards
please.
if you want to know. Okay, so we have a question here from Brian. Did you evaluate recovery rates as well as survival rates for the various pesticide beneficial pairs?
Right, Brian. Well, in this case, we only look at survival rate. We did not look at recovery rates at all. It is very important for us to think about recovery rate in the field study, definitely. And unfortunately, in this particular study, we kind of did everything in the lab, make it a lot easier to get some of the preliminary data, so that it can inform us of what we want to look at in the field in the future. But definitely we'll be looking at putting what we learned today into the field in future study, just to see what we're seeing in the lab. Is it also panning out in the field as well?
Okay, and another question from Brian here. Sulfur is also widely used in organic production as an acaricide and fungicide. That might also be something to consider for future experiments.
Excellent point, Brian. And we did not look at sulfur in this particular case, but we actually have quite a bit of information about using sulfur and how they whether they're compatible with biological control agents or not. Unfortunately, for the data that I have on hand, a lot of them are suggesting that sulfur is not that great when it comes to controlling come to compatibility with biocontrol agents. Sulfur has still been used quite a bit uh, for insects and for mites and insects, and also fungal disease suppression in greenhouses, which is a system I work with a lot. And we typically do not recommend sulfur at all if we're using biological control, because we know by applying sulfur, it doesn't matter whether we burn it or we dust it or we spray it, doesn't matter. When we use that, oftentimes it will just wipe out our biocontrol agent population. So I would definitely recommend against that.
Okay, thanks. We have another question from Wei Yang here about whether or not you've seen any resistance when using Entrust.
Right. So, well, when it talks about resistance, oftentimes it's pretty species specific. So at least in the southern vegetable system, and we are definitely seeing quite a bit of reduced sensitivity. I can't really quite say whether it's resistant or not, but we do see that Entrust or spinosad is not working that great against some of the pest species that we use it a lot on. For example, diamondback moth. At least in South Carolina system, we're seeing a lot of failure with using spinosad for controlling diamondback moth. And we think there's some resistance issue there, but we never really have the time or the resource to really do a detailed study on really confirming that it is resistant or whether it's just reduced sensitivity. But spinosad or Entrust, we have used it so much in our system, it would not surprise me that there are actually resistant issues. And another system that I work with quite a bit is ornamental plant system. And we know that spinosad, in that case, the trait name is conserved, actually have quite a bit of resistant problem in thrips population in greenhouses. A lot of growers are already reporting that thrips using conserve or spinosad to control thrips, they are not getting the effect that efficacy that they usually see in the past. So yeah, I think there will be some resistant issue out there with spinosad.
Okay, if anybody else has any other questions, I'm just going to leave another minute or so for you to type those in. This is a good opportunity to ask your questions since JC is right here. I just also wanted to mention while we're waiting that we're going to be having some more webinars from this project, different aspects of this project in the coming month. There's 2 of them that I'm going to put on the schedule at some point within the coming week in May. So I don't know if you want to talk about those, JC, at all.
Sure, I can talk about it.
Mhm.
I think we're going to have 2 webinars in May, and one of them is from Tom Bilbo here at Clemson University, and also Jim Walkabout from NC State. I believe that's early May. They're going to talk about how you can build your own predatory mite system, so that you don't always have to buy them. You can just build your own, and how you can use that predatory mite system in tomato production. And some of the work they have done in North Carolina basically showing that Phytoceres persimilis, you can rear them pretty easily on your own. And in fact, you can rear them that is more adapted to the tomato system so that you can use them directly in the tomato field. And later on in May, Babu Srinivasan from University of Georgia is going to talk about a relationship between, biological control and viral disease transmission. Basically, it's looking at whether we can use biocontrol to really reduce the pest population and therefore reduce the viral disease incidence, particularly from whitefly.
Okay, great. Yeah, okay. It doesn't look like we're getting any more questions. So I'd just like to thank everybody for joining us today. And thank you so much, JC, for giving another presentation. And as I mentioned, we're going to put the slides and the recording up on the eOrganic website. To find them, you just Google webinars by eOrganic, and then you can see our whole list. And then you can find the recordings and any other supplementary information on the page with the recording. So thank you very much again, and we'll see you all soon. We have some more webinars coming up this month on other topics. There's one on grain marketing, which I'm going to put on the schedule today. So thanks very much. Bye.
Thanks, everybody.